will be our presenter today. Dr. Mariana Diniz graduated in biological sciences and obtained her PhD from University of São Paulo, studying vaccines against viral infections involved in cancer under the supervision of Professor Luiz Carlos de Souza Ferreira. She completed her postdoctoral training, spending different periods of time at the Biological Science Institute at USP, at the Hôpital Européen Georges Pompidou, Paris, and at the University College London. Currently, she is an associate researcher in the Division of Infection and Immunity at the Institute of Infection, Immunity and Transplantation at University College London, where she was a recipient of a grant from the Therapeutic Innovation Network Pilot Data Fund. Her studies focus on how cell therapy approaches can enhance the immune response against liver cancer and viral hepatitis. And today, she will be talking to us about harnessing tissue-resident T cells in viral infections and cancer. So, Dr. Mariana, thank you again for accepting our invitation, and the floor is yours.
Thank you, Bruno. Thank you so much for the invite, and thanks for everyone that joined. It's a very nice to be speaking to you all and seeing some rooms that I can see from the ICB, where I started my research journey. So yeah, really a great pleasure to be talking to all of you today. So what I've had planned for today is, as Bruno mentioned, to talk about tissue-resident T cells in viral infections and cancers. But of course, this is a very broad and ambitious title. So for today, what I had planned is to focus more specifically on CD8 T cells, but we can later discuss about CD4 T cells also, and also focus on the liver as a site for differentiation of these tissue-resident T cells. And within the liver, as Bruno touched upon, my lab focused on chronic hepatitis B and liver cancer, particularly hepatocellular carcinoma. So we'll be talking about tissue-resident T cells in the context of these diseases. And then if we have time left, I'll also talk a bit about some work that I have been doing on SARS-CoV-2, looking at tissue-resident T cells in the lungs then. So just to start, and very briefly talking about resident T cells differentiation, after priming in the lymph node, we know that the T cells develop into different subsets of effector cells and memory precursors. And some of these subsets have the ability to migrate to tissues and respond to signals from inflamed and infected tissues. And then within the tissues, after they migrate, they can then develop a more mature tissue-resident phenotype. So different signals from the tissue, for instance, IL-15 or TGF-beta, can enhance the development of these tissue-resident T cells. They usually express CD69 and CD103 for CD8 T cells, but really depending on the tissue or the organ that these resident cells are developing, they will express different receptors and different cytokine receptors that also contribute to their migration, but also retention in the tissues. And what are really hallmarks of tissue-resident T cells is that they become unresponsive to signals that promote homing into blood. So they really retain in the tissues, and they don't circulate through secondary lymphoid organs anymore. There is now a broad description of tissue-resident T cells in several organs and tissues across the body, and they are thought to be posed with a quick response, and very interesting then at the site of infection. So the advantage of have a strong TRM response is that they provide these really frontline response at the site of infection. They are shown to be responsive more quickly than other memory subsets, for instance. They retain a autonomous cytotoxic potential, showing a constitutive expression of granzyme B and perforin, and they have this capacity to remain in the tissues for really prolonged time without the requirement of constant antigen exposure. So the environmental cues from the tissues can sustain their longevity. And also I'll start focusing now a bit on the liver, as I mentioned, for my lab's interest in HBV infection and liver cancer. And particularly chronic HBV infection remains as a major cause of death worldwide. So despite the existence of a prophylactic vaccine for chronic hepatitis B since the early 80s, still there are around 300 million people chronically infected with HBV, corresponding to more than 3% of the global population. And the main concern with chronic hepatitis B infection is this progressive disease state leading to chronic liver inflammation, liver cirrhosis, which is a major health burden, and the possibility that this also progress to liver cancer. And then within the primary liver cancers, HCC or hepatocellular carcinoma is the biggest form of primary liver cancer, the most frequent form. And still like around 800,000 of annual deaths are attributable to HBV-related cirrhosis, and especially HCC. And what we see with different chronic infections or other cancers where the death toll is decreasing, with HCC is the opposite. Death rates continue to increase and are estimated to increase further in the following years. Considering chronic hepatitis B infection, there is still a major urgent need for novel therapies, because current therapies require long-term treatment and they control, but they don't eradicate the virus. So there isn't a current curative treatment for hepatitis B, for chronic hepatitis B. With acute hepatitis B, the scenario is a bit different, because acute hepatitis B, which happens usually when infection occurs in adults, is rapidly resolved by the host, and control of infection is followed by a strong activation of T cells and B cells. So there's antibody production and detection of antigen-specific T cells for several HBV antigens. And these, along with other evidences, the fact that HBV resolution is followed by induction of immune system suggests that if we can also develop a immunotherapy or other measures to boost the immune system, this could also help to control chronic hepatitis B and liver cancer. And this could go along with current therapies that are now based on antiviral treatments. So the rationale that immunotherapies could have a potential role in chronic hepatitis B then drives the focus to try to understand better what's happening with immune cells in the liver. And the liver is an organ that is designed to be tolerogenic, so it receives blood from the gut, and this blood is filled with food antigens and other antigens from commensal bacteria. So the liver is designed to suppress immune responses rather than induce like strong inflammation or pro-inflammatory signals. So with this, the whole environment in the liver is designed to limit T cell function. So it's a hypoxic environment and has lots of suppressive cytokines like IL-10, TGF beta, and several immune populations that are also suppressive. So it's filled with granulocytic myeloid-derived suppressor cells or suppressive neutrophils, and other populations like Tregs and regulatory B cells. And however, we do see in the liver populations that can contribute to a efficient adaptive immune response, and it can be remain some functionality. So some years ago, our group drove attention to liver resident CD8 T cells, and we were actually the first group to describe the presence of these cells in the human liver a few years ago. And what we found is that in the liver, there is this population of memory CD8 T cells that express CD103 and CD69 that are present in the liver at different frequencies and are absent in blood. And what we also saw is that compared to healthy livers or livers from uninfected individuals, the percentage of these TRM population is increased in liver from donors infected with chronic hepatitis B. And furthermore, we also saw that besides the expression of these hallmarks of TRM, these cells have a distinct transcriptional phenotype. They express BLIMP1, which is also now considered a hallmark of tissue-resident T cells. What was also important and relevant from this work is that in the liver of patients with chronic hepatitis B, where lower viremia was detected, there was a higher, lower viremia was detected here on the left. There was a higher abundance of tissue-resident T cells. And this suggested a potential for at least this association that TRMs could help to control the HBV viremia in the infected liver. So with this work, we then wanted to try strategies to further harness the TRM functionality to then potentially improve their role in controlling chronic hepatitis B infection. So one of the strategies that are being intensely investigated for chronic hepatitis B in the immunotherapy setting is the possibility to develop a therapeutic vaccine that could potentially rescue already existing T cells that are circulating or that are in the liver, but also that could induce de novo T cell responses. And the issue is in chronic hepatitis B, as I mentioned before, there is a whole environment in the liver that promotes the dysfunction of T cells. So there are cells that are deleting antigen-specific T cells, like NK cells. There is this long-term antigen exposure, and that ends up promoting exhaustion or dysfunction of T cells in particular. There are regulatory cells and suppressive cytokine milieu. So it's really difficult to, so the therapeutic vaccines tested in the clinic so far have really failed to promote these, the boosting of these antigen-specific T cells and induce these more pro-inflammatory response, ideally with lots of antigen specific antiviral T cells producing cytokines like interferon gamma, TNF, and IL-2 to then definitely control viremia. So there is now this consensus that therapeutic vaccines on their own are not going to be enough to induce a sufficient control of chronic hepatitis B and overcome this T cell dysfunction. And different strategies are now investigated to try to be combined with therapeutic vaccine to actually harness a antiviral T cell response, particularly within the liver. So to start testing therapeutic vaccines and what we could combine with them to then potentially induce a strong T cell response in the liver, we then started using a animal model of chronic hepatitis B. So hepatitis B virus don't infect mice, they only infect humans and chimpanzees. So what we're using in the lab is a surrogate model of infection where we use an adenoviral vector that encodes the whole HBV genome, and we transfer these adenoviral intravenously to the mice. And then it has tropism to the liver, infects the hepatocytes, and the HBV genome is actually only expressed in the hepatocytes. And with this model, we can detect expression of HBV proteins for over 1 year in the liver of these mice. So they don't eliminate the infection, and we're able to induce this persistent infection. And in this model, we are testing a therapeutic vaccine that is actually now in clinical trials. It's based on a ChAdOx vector, so chimpanzee adenoviral vector, the same that was used by in the SARS-CoV-2 vaccine from AstraZeneca. And this ChAdOx virus encodes three major antigens from hepatitis B, so core, polymerase, and surface antigen. And we give this vaccine intramuscularly in a single dose regimen. And then to try to see if we could increase vaccine response, particularly focusing on try to increase the development or differentiation of tissue-resident T cells, we combine this vaccine with administration of IL-15 fused to IL-15 receptor. And the idea of giving IL-15 is because, as I mentioned, it's one of the cytokines best established to support TRM differentiation and also retention of TRMs in the tissues. And we're giving IL-15 here together with the receptor alpha to avoid the need for a APC for trans presentation. So just to highlight some of the results that we've found in this project that it recently started in the lab, we can see that after immunization, there is a expansion of CD8 T cells in the liver, but we see that IL-15 supports a further either proliferation or infiltration of global CD8 T cells to the liver. We see that they are actually also proliferating, not just coming from the liver, from the circulation, showing here by increased frequency of Ki-67 positive CD8 T cells. And when we then start looking at antigen-specific cells, so here using tetramer staining, focusing on one of HBV antigens, so HBV envelope protein, which is the surface protein, we can see that in the group that receive IL-15, we have a higher percentage of the CD8 T cells that can actually recognize surface antigen from HBV. So, and we also see that we have a stronger differentiation of tissue-resident T cells in within the antigen-specific CD8 T cells. And in mice, there is a difference from humans here to define CD8 resident T cells. In mice, they express CXR6 and 69. And what we saw that when we're giving a vaccine alone, here in the bar in the middle, there is a trend to a control in viremia, in HBV viremia, compared to untreated mice. But we only see a significant decline in viremia when the vaccine is combined to IL-15. So also suggesting that potentially inducing more TRMs can contribute to improved control of viremia. We are following now in this project, and we really want to try to dissect more the role of TRM compared to other CD8 T cell subsets in controlling the viral infection. So we're trying to isolate TRMs and other effector memory populations and see if this, and transfer these to mice and see if it can impact viremia. But then in the meantime, we had other questions that we tried to address. So besides doing this more direct induction of TRM using cytokines, we were also wondering what else is limiting the TRM functionality to exert this more pronounced antiviral role in the liver, because we can detect them in the liver and we see that they are associated with improved viral control. But what is impeding that they have a more pronounced activity? And in particularly, we were interested to see if other cells in the liver were inhibiting TRM function or differentiation. And then in this context, looking at other cell interactions, we then focus our attention on NK cells. And although this is counterintuitive, or can seem counterintuitive at the beginning, thinking of NK cells as a cell that would suppress T cells, our work from our lab had found some cues to why that would be the case. So I say counterintuitive because NK cells are well known for their antiviral functions and antitumoral function as well. And they can also support T cell function. So besides their direct cytotoxic activity, they produce cytokines such as interferon gamma that can boost T cell responses, including CD8 T cells. However, in hepatitis B, in chronic hepatitis B, our group has found that the NK cells can actually limit CD8 T cell responses. And they are doing this directly. They have the capacity to actually kill CD8 T cells via TRAIL or NKG2D. And this ability of NK cells to kill CD8 T cells in chronic hepatitis B is linked to a subset of NK cells that is also resident in the liver, called liver resident NK cells. So then, having seen this before in our group in chronic hepatitis B in humans, we wanted to then try to address if the NK cells could also limit T cell responses to therapeutic vaccination in HBV, and then further if we could modulate or manipulate this interaction to try to unleash CD8 T cells from the control of NK cells. So using the same animal model that I described before, and actually same vaccine, we now tested NK depletion just before the vaccine to try to investigate if the NK cells had any influence in the T cell responses that was being induced by the vaccination. So in this case here, we see that the vaccine alone can increase the functionality of CD8 T cells. In this slide, we're looking at interferon gamma production, after peptide stimulation using peptide pools covering HBV antigens. And we see that the NK cells, that the vaccine can induce a interferon gamma response by the CD8 T cells to different antigens of HBV, on the left surface antigen and on the right polymerase antigen. But we see that when we combine NK depletion with the vaccine, which is the bar on the right, we see a further enhancement on CD8 T cell functionality to both antigens also. So suggesting that in the absence of NK cells, the vaccine could induce stronger T cell cytokine production. We also saw that the NK cells were limiting not just T cell functionality, but also their expansion, or the expansion of antigen-specific CD8 T cells. So here, using again MHC multimerase targeting surface antigen, we could detect a very mild expansion of antigen-specific CD8 T cells after vaccination, but we could detect a significant increase in the proliferation of antigen-specific CD8 T cells when the vaccine was given in mice that had NK cell depleted. And when we looked at the different memory differentiation, we saw that in fact, the subset of tissue-resident T cells that express 69 and CXCR6 was also being increased in the absence of NK cells when combined with the therapeutic vaccine. Together with other results that we found in this project, we saw here that NK cells are exerting a dominant negative regulatory effect on T cell responses. So they're limiting T cell responses. And in fact, we saw that this is restricted to the HBV infected liver. So we didn't see these differences in circulating T cells or T cells in secondary lymphoid organs. And the fact that this is restricted to the liver then made us turn again our eyes to the liver and focus on populations that are resident in the liver. So because we were seeing this, the important role for NK cells in inhibiting T cell responses, we then wanted to test if this was being induced by NK cells that were infiltrating the liver, or if they were being induced by liver-resident NK cells. So in mice, we can distinguish conventional or liver-resident NK cells based on the expression of DX5 or CD49A, with the liver-resident NK cells expressing CD49A. So what we've tested now was sorting either conventional or liver-resident NK cells and transferring these to the mice before giving the vaccine. And then we looked for what this would cause through the induction of CD8 T cell responses. And one example of a result that we found was that when we transferred liver-resident NK cells, the T cell capacity to produce cytokines such as interferon gamma to different HBV antigens was lower compared to mice that received vaccine only. And we saw that when we transferred conventional NK cells, the response was either the same or even higher than compared to vaccination alone. So showing that really the population of NK cells that was contributing to limiting CD8 T cell responses was more likely of the liver-resident NK cells. And then we were interested to try to find if there was a targetable pathway mediating the liver-resident NK cell regulation of T cells that we could then use to try to boost T cell responses. So we looked for several potential receptors on the NK cells and their ligands on the T cells, pathways that we had described already in chronic hepatitis B, such as trail-mediated NKG2D, or pathways that were described in other diseases like NKp46, but really a broad spectrum of potential markers. And what we saw that was more striking was that in the chronic infected mice, so in the HBV infected mice, we saw that the liver resident NK cells were enhancing expression of PD-L1 on their surface, and this was not happening on conventional NK cells, so NK cells that were infiltrating in the liver, and was also not happening on circulating NK cells in secondary lymphoid organs. So this was a potential pathway that we wanted to further explore as relevant for liver resident NK cell regulation. We then tried to see on the other side of the CD8 T cells, then how was then the PD-1 expression on CD8 cells, particularly on HBV specific CD8 T cells. And we saw that in the liver of the HBV infected mice, PD-1 is highly expressed on antigen-specific CD8 T cells, and we didn't see this upregulation of PD-1 on splenic CD8 T cells, for instance. So this was showing that the antigen-specific cells, and particularly this is true for tissue-resident T cells, there is this really high expression of PD-1 that could make them susceptible for regulation of liver-resident NK cells via PD-L1 on the other side. And we further dissect this pathway for NK cell regulation. So we combine the therapeutic vaccine with anti-PD-L1 or anti-PD-1 antibodies, and we saw that when we're combining the vaccine with these PD-1 blocked, we had similar effects as we had seen with NK depletion. So this redundancy between the two pathways was also reinforcing that the NK cells could be acting via PD-L1. And we also transferred to mice PD-1 knockout CD8 T cells, and we saw that these were not susceptible to NK cell regulation. So again, confirming that the PD-L1 pathway could be relevant for T cell regulation by NK cells. And then to confirm that this had a translational relevance, that this was not just something that we could see in mice. We also looked at expression of PD-L1 on liver-resident human NK cells, and we saw that PD-L1 is enhanced in the liver-resident NK cells compared to the conventional or circulating NK cells, and particularly it's particularly higher on HBV-infected cells. And we also see that on CD8 T cells in the liver, there is a much higher PD-1 expression on the CD8 cells. So again, showing that this pathway could be also relevant in humans. And so just to summarize this part, we've seen that HBV infection drives a PD-L1 dependent liver NK cell regulation of vaccine-induced liver-resident T cells, and then targeting the PD-1 axis could avoid NK cell negative regulation of T cells and potentially improve responses to therapeutic vaccination. Because one of the main concerns or main health burdens related to chronic hepatitis B is the potential to induce liver cancer, we then wanted to see if this pathway was also relevant in liver cancer. So then a PhD student in the lab, Stephanie, she set off to try to investigate these cells and these pathways in HCC. So for this, we established a mouse model of orthotopic HCC, where we inject in the mouse liver using a ultrasound guided system hepatoma cells, and we engineer these hepatoma cells to express HBV surface antigen, using surface antigen here as a model tumor antigen, and it's a tumor antigen that is commonly expressed in HBV related HCC. So what Steph then started doing is, after establishing these tumors in the mouse liver, she gave the same vaccine that we were using before in hepatitis B, and first started again depleting NK cells and seeing what the responses, and try to see what the responses would look like. And here she saw that when we look at the spleen of mice, we don't see a difference in the function of CD8 T cells, here shown by production of interferon and TNF alpha, when the vaccine is given alone, or in NK cell depleted mice. But she did see that when she looks at CD8 T cells infiltrating the tumor, there is enhancement on the frequency of cytokine producing CD8 T cells when NK depletion is combined with vaccination. So again, suggesting that the NK cells could limit responses to vaccine. And when we looked at the tumor control, so here measured by the tumor weight at the endpoint of the experiment, again, she saw that the NK cells were impacting the vaccine responses. So only in the group where vaccine is combined with NK depletion, we see that there is a significant control of tumor growth. So again, showing that NK cells have a predominantly inhibitory role in the vaccine responses in this setting. When then we looked at the presence of tissue-resident CD8 T cells in the tumors, we see that they are present at significant frequencies. And we also see that, specifically looking at the resident CD8 T cells, they express higher frequencies of PD-1 compared to the circulating T cells that infiltrate the tumor. And also, then looking at the NK cells on the other side, we see that in the tumor, they also upregulate PD-L1 compared to circulating NK cells. To again try to look at the translational relevance of this pathway now in human HCC, we saw that from cells extracted from patient tumors, there is a higher proportion of PD-1 expressing CD8 T cells infiltrating the tumors compared to blood in patients with HCC. And like what we saw in mice, we saw that the resident CD8 T cells are the ones where PD-1 is enriched compared to circulating CD8 T cells. And on the other side of the cell-to-cell interaction, we also found that in human HCC, there's a higher frequency of PD-L1 on the NK cells infiltrating the tumor compared to to blood. And then again, the liver resident NK cells are the ones that express higher amounts of these ligand. And what we also saw is that we have some preliminary data where we're starting to look at resections from HCC, and we are being able to identify NK cells and CD8 T cells in close contact. So suggesting that these cell-to-cell interaction between NK cells and T cells within the tumor is also possible and a likely mechanism of suppression. We've seen then that the NK cells, especially the ones found intratumorally have the potential to limit vaccine-induced CD8 T cells also in HCC. And further work that we're doing now will be on dissecting now the role for the PD-1 axis more specifically in HCC. And we then wanted to try other strategies to avoid these NK cell regulation of T cells via the PD-L1 axis, and but also taking advantage of the NK cell beneficial effects, because as we discussed, the NK cells have these intrinsic anti-tumoral effects. So we wanted to try to find a way to still harness the NK cell anti-tumoral effect, but then avoid their limitation of T cells. I'm running short on time, so I'll go very quickly here. What we then wanted to do is move away from therapeutic vaccine, but then in HCC, think about a adoptive cell therapy. And we then focus on the NK cells, which as adoptive cell therapy show lower toxicity compared to T cells. And also they bypass the need for autologous cells, which is a major advantage in cancer. And we were then using cytokine activation to harness the NK cell direct cytotoxicity, and also cytokine activation makes NK cells produce lots of cytokines like interferon gamma, which then, as I mentioned before, helps to increase T cell function. And we had also seen that in HBV, HCC, but also with cytokine activation, there is a big upregulation of PD-L1 on the NK cells that can limit T cell function. So what we plan to do is then to engineer NK cells to make them produce and secrete anti-PD-L1 antibody. And the idea is that this would bind PD-L1 on the surface of the NK cells, but also block PD-L1 on other cells in the tumor microenvironment. And with this, would then limit their capacity to limit T cell function, which the T cells express high PD-1. And another limitation of NK cell as adoptive cell therapy is their poor tumor infiltration capacity. So we also aimed to engineer these NK cells to express CXCR6. CXCR6 is a receptor largely expressed by liver-resident NK cells and induce their homing to the liver, but also to the tumor in HCC, driving their migration to the ligand CXCL16. So the idea here was that we would engineer NK cells to home and then be retained within HCC, and then they could have their direct anti-tumor function and serve as a local source of anti-PD-L1 antibody to unleash T cells, particularly resident T cells. And then here I was just going to show that we can successfully transduce NK cells with lentiviral vectors, so we can induce expression of CXCR6. And the NK cells that express CXCR6, they have an increased capacity to home towards tumors. So here in this transwell assay, we see NK cells towards a layer of hepatoma cells, and we see that the NK cells transduced with CXCR6, with the transgene of interest, they can migrate more efficiently and actually target the tumor cells. And the tumor cell lysis is abrogated in the presence of anti-CXL16, so showing that the migration is actually driven by this ligand. And also in assays with tumor cell spheroids, we see that CXCR6 expression enhances the capacity of NK cells to induce tumor cell lysis. We also confirmed that the NK cells transduced with the vector of interest can produce PD-L1, and we see that when the NK cells are transduced with a control vector, there is a high detection of PD-L1 using flow cytometry. And as we expect, when the NK cells are producing anti-PD-L1 antibody, this would block the ligand on their surface and avoid staining with fluorescently labeled antibodies. So that's why when we have the NK cells transduced with the transgene, we have this low detection of PD-L1, suggesting functional blockade by the antibodies that they are releasing. And just the last thing, when we use the NK cells combined with CD8 T cells that are antigen specific for the tumor cells, we can see that when the NK cells are transduced with the transgene of interest and producing PD-L1, now the CD8 T cells are able to produce superior tumor cell lysis, suggesting that the PD-L1 blocking by the NK cells can increase the CD8 T cell function. So just to summarize then this part, we see that cytokine activated NK cells can be engineered to express CXCR6 and secrete anti-PD-L1 antibody and have the potential to home to the tumor and potential to boost intratumoral antigen-specific T cells. And the therapeutic implications of what I've tried to discuss here today is that boosting tissue-resident T cells can support viral and cancer control, and also that TRM induction directly via cytokines or targeting indirect pathways such as PD-L1 in chronic hepatitis B and liver cancer can support their development and function. So I think I'll stop here. We won't have time to go to the other part of the presentation. And yeah, happy to take any questions.
All right, thank you very much. As always, we're going to open first for questions from the students.